Hello and welcome to another video lecture from Mr. Watkins. Today we're going to focus on protein synthesis. Specifically, we're going to look at the central dogma, we're going to look at transcription, and we're going to look at translation. Translation will have its own video, so consider this part one of a two-part lecture. So, what is the central dogma? The central dogma of biology is basically describing how information carried on molecules flows through the biological system. Specifically, how the information that is stored on DNA is transferred to RNA, messenger RNA in our case, then sent to a ribosome so that it can be made into a protein. So you may hear DNA makes RNA makes a protein. The big thing that Francis Crick said when he went through and basically coined this is that this process of information transfer is one way, and so that when it gets to a protein, it doesn't go back the other way. Once that protein is done, it's done. The information doesn't travel backwards. So let's start talking about the two processes here that we need to go over. One of them is transcription, and this is the first part of that flow of information. The other one is translation. Transcription is basically just the copying of the DNA nucleotide sequence for the gene into a messenger RNA sequence, so it'd be RNA nucleotides for that gene. It makes a copy. This occurs in eukaryotic cells in the nucleus. Translation is taking that mRNA and converting the nucleotide sequence into an amino acid sequence. So we basically convert it into a protein. And that's done in a ribosome. And in a eukaryotic cell and prokaryotic, it's also done in the cytoplasm. So let's get into the details of transcription. Now in this video, I'm not going to get into all the details. I'm only going to give you an overview. If you'd like all the details, the nitty-gritty, I have placed notes in Canvas that give you all the details on every piece that you should know, and I strongly recommend that you look at that. All right, so let's look here at transcription. So transcription has three parts to it. Okay, these may be familiar to you because we've discussed some of these terms before. The first is initiation. Really, the only thing that you need to know here is that this is the beginning of the process. And the beginning of this process is that the RNA polymerase, it has some function that's similar to DNA polymerase. RNA polymerase has to find a special region called a promoter region that is located on the DNA that says, here's where you need to start to copy the sequence of DNA and make that immature mRNA, that mRNA that's going to be hopefully later processed and used to build a protein. This promoter region just says start here. Okay, you don't need to know a lot of the details about how all that works. Okay, now the one thing that you do need to be aware of is that that piece of DNA strand is a template. It's not destroyed, it's just copied. Okay, so we sometimes refer to that as the antisense strand or the minus strand or the non-coding strand. So you need to know those vocabulary words. Again, I'll point you to the notes that I placed on Canvas for you. Okay, so RNA polymerase finds the spot it needs to start the process at that promoter region. RNA polymerase can unwind, unzip, add the nucleotides and to make that copy, produce that mRNA, and then the DNA will zip right back up in all one nice big package. So the second step is elongation, and that elongation is the process that I just described. It's the RNA polymerase adding the complementary RNA nucleotides to form what we call an RNA transcript. That transcript, you probably have heard it being called as mRNA. Okay, it is mRNA, but we typically refer to it as a transcript, meaning it was copied or transcribed off of the DNA. And so the RNA polymerase continues to make that copy, putting the correct nucleotides, just like we talked about with Chargaff's rule. Remember, there's no thymine, it is uracil, and it makes a copy until it gets to the last stage, which is called termination. Okay, so in termination, there's a specific set of proteins that are at the end of the sequence of DNA nucleotides, that gene, and these proteins are attached down there, and they basically say, stop. They tell it to stop, and what happens is that the RNA polymerase falls off or comes off of the DNA sequence, and the mRNA that was made, which is now what we call an immature or pre-RNA, is released and floating there, either in the cytoplasm if it's a prokaryotic cell or in the nucleus with a eukaryotic cell. So what has to happen next is we need to change that pre-mRNA or that immature mRNA and have that processed to a mature mRNA so it can be sent to a ribosome and then be translated into a protein. So what are the three things, and there are three things that you have to know that happen with the processing of that mRNA. And these three things all use enzymes to get that to work. So one of the first things you need to be aware of is the removal of what we call interons. Interons are what's called intervening sequences. See, the DNA copy, or the DNA, sequence has all those nucleotides in it, but not all the nucleotides are actually necessary to make that protein. And so some of the nucleotides that are found don't actually get expressed into a protein. And so what we need to do is take that pre-mRNA and convert that, or excise, which means to cut out, those sequences that we call that are intervening. They are not going to be expressed. And what we want to have left are exons, which are the sequences that are left over after we've pulled out the interons, that will be used to make the actual protein. So kind of what I want you to think about is the fact that we may have this chunk of DNA, and then it gets copied to mRNA, and some of the pieces of it get copied over, are going to be used later in making that protein, and some of them sequence or not. And so we want to remove these interons, and there's enzymes. You don't have to know the names of those. We want to remove those enzymes, so what we get left over is just the exon, the pieces that are needed, or the sequence that's needed to build that protein. Okay. The second thing you need to be aware of is that we have to also help stabilize the mRNA and help keep it from being attacked by other molecules that may be floating around in that cytoplasm. So enzymes come in and they add a poly-A tail. Basically, all this is a series of adenine nucleotides, and it's a bunch. It could be 100, 200, or even more that are added at the end, the 3' end of the mRNA molecule. And basically, they're there to protect that mRNA so that it doesn't degrade as it moves from the nucleus. In eukaryotes, moves from the nucleus out to the cytoplasm, and some molecules may attach it or try to attack it. Same thing in prokaryotes, except of course there's no nucleus, and so it's helped protect it from being degraded by enzymes and other molecules that would be there in the cytoplasm. And then the third thing that you need to know is what's called a GTP cap. It also can be referred to as a 5' cap. your particular course would prefer that you use GTP-CAP, and it is just basically guanosine triphosphate. Similar to adenine triphosphate, ATP, this is just GTP. And its purpose is to, again, protect the molecule, the RNA molecule, so that it doesn't have any problems with other molecules that may be there, other enzymes that may be there to try to degrade it. Once these three things are done, we end up having a mature mRNA that is ready to be translated into the protein. If you have any other questions about what's going on, please rewatch the video. There's lots of Google sites that actually show this in real time for you. I also have put some links in as references for you in Canvas. And again, please look over the notes that I provided that give you all the details that you need.